everyone. Thanks for joining this live webinar by World Molecular Imaging Society. Title of our talk today is Oxygen-Enhanced Electron Paramagnetic Resonance Imaging for Evaluation of Radiotherapy in Preclinical Tumor Models. Before we go to the webinar, there are a few announcements I want to make. Oops. The Prague meeting late breaking abstract submission is still open, so we hope to see you in Prague. Please submit your abstract as soon as possible. We also have a special issue on the subject of EPR oxygen imaging and its application to biomedical sciences for molecular imaging and biology journal. And if you're working in the field, we look forward to receiving your submission for the journal for this special issue. And after the webinar, we will all be at social launch. Marty, I, and everyone who's interested in Q&A and chatting, please stay. So don't log out from Zoom, but you can join to social launch. And with that, it's my pleasure to introduce Dr. Marty Pagel, whom I know for some time now. Dr. Pagel is a professor in Department of Cancer System Imaging and Imaging Physics at University of Texas MD Anderson Cancer Center. His research in academia and industry has evolved from chemistry to biomedical engineering to molecular imaging. And his current research employs a variety of imaging modalities to interrogate biomarkers of the tumor microenvironment. He has developed these methods for preclinical and clinical imaging, especially to evaluate cancer therapies. And Dr. Pagel has held many leadership positions in the molecular imaging research community, including roles in scientific societies, grant review panels, and journals focused on molecular imaging. And we look forward to your webinar, Marty, and the floor is yours.
Thank you so much. I'll share my slides. I'll share. So hopefully you can see the first slide here, Marignani.
Yes.
Okay, great. And I really appreciate the introduction, Marignani. It's always great to partner with you to talk about new science. And so I'm excited to be here here with others from the WMIS and our molecular imaging community. And I'd like to describe a new type of molecular imaging, oxygen-enhanced electron paramagnetic resonance imaging, sometimes just referred to as EPRI. I'll often use that term. And this is to evaluate radiotherapy in preclinical tumor models. And of course, I'm here at the MD Anderson Cancer Center. So to begin with, just have some conflicts of interest, work with a number of companies, including O2M Technologies, which will be part of this presentation. I'd like to first start with a bit of a philosophical view. I mean, why are we here? Why are we doing molecular imaging? And what really drives me to get up in the morning and get to the lab? Well, this is our value proposition. I think we need to focus on treatment decisions, make very specific quantitative measurements. And then, of course, this is molecular imaging. And I'd like to highlight that with just two schematics here. There are many people on Earth, right? And yet not all people are patients until they're initially diagnosed and they undergo treatment, hopefully remission and hopefully not recurrence. And so I really think that there is an opportunity for molecular imaging mainly to predict whether a treatment will work just prior to applying that treatment and also early response after that treatment. Now, certainly molecular imaging could be used as a screening technique, but I would argue that really the value is a ratio of benefit to cost. And we are a costly method. Molecular imaging is more costly than regular non-invasive imaging. And as we know, radiology departments are costly relative to blood sampling and so on. So if it were very costly, well, then we have to bring great benefit. And I think that benefit will be really for those patients who are undergoing treatment. And so I think the EPRI method is a great technique for looking at this prediction and early response, as I'll show in a moment. So that I'm kind of setting this presentation up with the strategy in mind. Also, I think we need to lower the cost of healthcare, but how are we going to do that? Are we going to lower the cost for those patients who are easy to treat? The often responders are easy to treat. The non-responders are not easy to treat and cost a lot more money. Well, you could try to lower the cost overall, but really, I think what we want to find are the non-responders and take them out of this paradigm of the ineffective treatment as soon as possible and get them onto effective treatment. Nobody wants to be a non-responder, of course. And as I've mentioned in the past, I think of non-responders as false positive patients. We thought they would respond, but they don't. This isn't a true definition of specificity, but false positive is related to specificity. And therefore, I think, again, we need to use molecular imaging to be more specific to identify or predict whether responders or a patient will be a responder or non-responder. And again, I think EPR imaging really can be used for this paradigm, especially for radiation oncology, as you'll see. All right, so with that in mind, our lab studies the tumor microenvironment, including oxygenation, and today we'll just focus on tumor hypoxia with EPR imaging. And so I won't talk about the other methods much, but I would like to start with a bit of a comparison with what has been done in the past to image hypoxia, and where does EPRI then fit on that landscape. So if you give me a few moments, I'd like to describe just a few other techniques. Many of us are very familiar with blood oxygen level dependent or BOLD MRI. That's been done for at least 20 years or more. The last ISMRM meeting had fabulous sessions on BOLD MRI, showing first how assumptions can be overcome, and also showing that there are remaining assumptions, and it is a complicated method to truly get right. And I do find in our own hands here at MD Anderson, there was a study done by colleagues, fortunately never fully published, where a very standard BOLD method applied at 7 Tesla with mouse models actually does have quite a lot of variation. And you can see other techniques also have a lot of variation, some not so much. And as I think we really do need a very precise molecular imaging method. Again, we want to be specific and quantitative. And to me, BOLD just doesn't quite get there. Now, I'm hopeful that those who are refining the BOLD technique can get us there, but there is a ways to go, especially at 7 Tesla, where there are some issues. Many of us are familiar with PET imaging agents. fMISO has been around for at least 20 years, never quite fully made it to the clinic due to the difficulty of washout from normoxic tissues. It tends to be a bit sticky. FASA is an improvement, more hydrophilic, and so it does wash out of tissues better, as shown in this example down here from a colleague, Seth Gammon and David Poinica-Werns here at MD Anderson, where you can look at the hypoxic status of a tumor and how that's affected by a drug using F18 FASA. And there's other agents out there as well that require some synthesis, but are under development as well. But I'd like to point out that you don't actually get a quantitative number from PET. You get either high, medium, or low retention if you do the experiment right. Yet, as I mentioned before, I want to be quantitative, to have a quantitative numerical scale to give to our radiologists or oncologists in order to understand whether their prediction to therapy or early response to therapy is actually correct. So to me, FASA gets there somewhat, but not quite. So then as a third method, and we've developed this in our own lab as well, but really I have to give credit to Ralph Mason and Sarah Bondic, who have developed oxygen-enhanced multispectral optoacoustic tomography, or MSOT. Here, a mouse will be imaged using the MSOT method with different frequencies of light that irradiate tissue. And then the ultrasound response can be mapped, and spectrum can be recorded at those different frequencies, and then unmixed to give you deoxy and oxyhemoglobin as a biomarker of tumor hypoxia. And that can be done with 2 different breathing gases. You can do an oxygen gas challenge to measure delta SO2 and get some information there as well. And there are limitations to photoacoustic imaging, or MSOT. It's only about 3 centimeters deep into the mouse or the patient, but yet it is a pretty intriguing technique that can give you a unique biomarker that can't be done with many other techniques very easily. So is this useful? Well, yeah, here's a study where radiosensitizers can actually improve immunotherapy. And we often think of radiosensitizers only for radiotherapy, but by reoxygenating a tumor in this particular case, and then applying a combination of antibodies, immune checkpoint blockade antibodies, or against those ICBs, then you do get a better tumor control in these mouse models. And so does that work with the this method? Well, yes and no. And that's why I wanted to highlight this method relative to EPR. I think it's a very interesting use of MSOT. But in this case, we're watching how the radiosensitizer agent actually releases oxygen from hemoglobin. So you get a counterintuitive decrease in percent SO2 as you oxygenate the tumor. You're deoxygenating the blood vessels, or the deoxygenating your hemoglobin. And therefore, you can't really tell what's going on in the tumor itself in that microenvironment, but you can see that there is some change in the blood vessels that does correlate with the response to immunotherapy. So kind of an indirect method. So we really, again, want to study the PO2, partial pressure of oxygen, within the tumor extravascular environment. Now, are we going to get there? Well, I think EPR has some merits there. So we want to study this O2 between the blood vessel wall and the tumor cell surface, right? That extracellular, extravascular environment where the immune cells are collecting and doing their job, or hopefully, right? Well, how does this EPR method work? We use an agent called OXO71, a deuterated version of another agent called OXO63 that's used for hyperpolarized C13MR spectroscopy. So some of us might be familiar with this structure. It has an unpaired electron right here attached to a carbon. This carbon has only 3 bonds to other carbons, so that 4th bond to a carbon is missing. Therefore, there is an unpaired electron. And I like this structure here where you can see this carbon is really buried. You can even see it even better buried in this ball and stick model here, or ball model. And so it certainly needs to be further developed and determined if it can be approved for clinical use, FDA approval, and so on. But at least there's a chance here, because that unpaired electron, being a free radical, is buried deep in the structure and therefore very protected from the rest of the body. So that I find very intriguing, that we have a relatively inert free radical. So that can be injected into a mouse. We administer intraperitoneally, so the agent lasts a while in the mouse, and you'll see in the experimental design in a moment, that's about 90 minutes. So I have to do an IP injection, but intravenous certainly works fine, and others have done that too. By the way, we're injecting at standard concentrations that are equivalent to what a standard MRI contrast agent would be, gadolinium-based, Dotaram or Magnavis that's injected in mice or in patients. So no, you don't need a much higher load, but it is equivalent to MRI in that regard. And here is a Jiva 25 instrument from O2M Technologies. I'm an MRI person by training, so when I was fortunate, actually it's a 25 millitesla magnet, not 250 millitesla. There was a typo there. And when I received the system from O2M, it's just astounding that you can do really great imaging on an electromagnet. I'd never done that before. And we're proud in the lab that relative to our MRI colleagues, we have the largest megahertz. Of course, it's electron MR frequency, but at 720 megahertz. And so we've enjoyed partnering with O2M Technologies to refine this technology for our particular use. Well, how do you measure PO2? Well, I'm sorry. This is really a very standard MR experiment, a T1 inversion recovery experiment that's done in MRI all the time, now applied to EPR, pulsed EPR imaging. So with a standard set of relaxation delays along the spin echo experiment, we can map out the T1 time. The T1 time is dependent on PO2 and has very little dependence on concentration. Enough that I consider it to be negligible for the studies that we're doing. R2, for example, the T2 time is concentration dependent on the agent, so that would not work as well. So again, as an MRI person, I find this really intriguing, that it's very easy to use, similar to an MRI scanner that I have in a back room here. And so we're able to put a mouse into a cradle that fits into our 7 Tesla Bruker MRI. And then we take that out of that room and into another bay that we've repurposed for the EPR machine. So it's a copper-lined room, a Faraday cage to protect radio frequencies from getting inside. And so then we slip the mouse inside the side of this magnet and right into the hole here, very accessible. Now, for years, I've struggle to get my arm inside those Brooker magnets and to position the mouse. And here the mouse is very accessible, which I enjoy. And then I think all imaging really needs outstanding physiological monitoring. And well, this instrument has a very nice system for maintaining the mouse at 37 degrees and measuring respiration as well, or monitoring that. There are fiducials within this cradle so that we can register the MRI and EPR images with no trouble. And the MRI is used for anatomical referencing, similar to PET MRI, if you will. So the MR analysis, EPR analysis is really pretty straightforward. It's a bit of an involved process to ensure that the registration is fine. But then after that, the calculation just takes off, no trouble at all. So one trained can do a calculation in about a half an hour. For me, it takes me about an hour. The students are still training me. And again, to further emphasize that this is really similar to standard small animal MRI, but just in a very different frequency regime for the electron. You have a standard coil called a resonator. You can tune. There's no match for these coils, no need, but very similar to how you would tune an MRI coil. There's coil tuning. You have optimized 90-degree pulse power. Then also look at pulse phase, partly to ensure that the spectrometer frequency is set on your sample. So for years, I've done a lot of manual shimming in my life. I've wasted many, many hours and lots of cups of coffee while shimming NMR and MRI scanners. I'm very excited to announce that you do not have to shim an EPR magnet. Now, the reason for that is that the line width is so broad, it wouldn't really matter if you shimmed, as so ridiculously large compared to MRI, and subtly changing the magnetic field will not change the line width. So it's kind of a, you might think of it as a detriment. I think of it as an advantage, so I don't have to shim. And here you can see a vial of the EPR agent, OXO 71, and a sagittal, coronal, and axial view, actually two vials that we put into the instrument. So you do get isotropic 3D imaging, anatomical images at 300 micron, and I believe we're at 660 micron when we do the PO2 mapping, just to improve the signal to noise and therefore contrast to noise and get a little bit more precise measurement. But 600 micron is great for measuring a tumor PO2. All right, actually, if I were to go back a couple of slides, there is one other point that I should mention that I've failed to before. The T1 time of this agent is 8 microseconds without oxygen, and it gets down to a microsecond with oxygen. Now, for those of us who do MRI and we think of T1 times being in the hundreds of milliseconds or even second range, that's astounding that we can measure T1 as short as 1 to 8 microseconds. But with the EPR instrument, it's very easy to do nanosecond type pulses and acquire the data very rapidly. So that's all been worked out for you to do 3D EPR imaging, T1 based imaging. So I like to joke that when I come from an MRI background, when I talk to those who do EPR imaging, it's kind of like talking to somebody from Australia. I can understand most of the words, but every once in a while, I'm just jolted by something really intriguing, like an 8 microsecond T1. Never thought that would be possible. All right, well, if we were to move on, here's a study that we did with this instrument to show the merits of measuring PO2. And we call this an EPRI MRI tumor oxygenation study, or oxygen enhanced, because we're going to change the amount of oxygen in the breathing gas. First, we anesthetize the mouse. We inject our agent intraperitoneally prior to doing any imaging. We allow that to circulate in the mouse so that by the time we get to the EPR imaging, it is circulated for at least 27 minutes and it started to accumulate in the tumor and will last for the entire duration of our experiment due to the IP injection. So we prep for MRI, do anatomical MRI to identify the location of the tumor relative to the fiducials. Then we prep for EPR. Again, just takes 10 to 12 minutes. Normally we're done after 10 minutes, and we're just waiting around a couple of minutes for consistency. So all of our mice start at 27 minutes. And here's a PO2 map. And well, I like to very affectionately call this a blobogram. It reminds me of PET imaging, where you see signal everywhere where the agent has gone. And you really don't have anatomical information. You only see where the free radical is, right? And here we can circle the tumor in the MR image and then overlay that on the EPR. We have a tolerance of about half a millimeter. It's about again, the size of a pixel within our PO2 map. And then we can change the gas, so wait 12 minutes for equilibration and then do that measurement again. We're also doing test retest in this study. So that way we can measure, well, what is the difference between the two scans for each of the 10 mice in the study? We're able to show that there's 2.6 torr for repeatability for both the 21% and 100% O2. So it's nice that it came out to be the same. And of course, if you look at the two scans done with medical grade air, 21% O2, there is no statistically significant difference to the order. Same for the oxygen. But if you do change the breathing gas, of course, you see a very different value of PO2 within the tumor. So we're going to use that to our advantage, that change in PO2 when changing gases. Kind of like using oxygen as yet another contrast agent. By blowing that into the mouse, you're adding an agent to the mouse. All right, so we have excellent repeatability. And in fact, as you may recall on a previous slide, I showed that MRI precision can be at best 15 to 20% for a lot of our common measurements, sometimes as bad as 40 or 50% imprecision. And I find that frustrating in small animal imaging. Here we've got test, retest, repeatability showing that it's on the order of about 10% of the PO2 measurement. And that is outstanding because I hardly ever see 10% precision in small animal imaging. PET imaging can get down to that level if you're very careful. Even optical imaging doesn't quite get below 10% very often, unless, again, you're very careful with your positioning of the mouse and so on. So this EPR instrument, I find, is very reliable. So we studied a Colo 357 model and an SU 8686 model. We were hoping that the Colo 357 model would be more hypoxic than the SU 8686 model. But to be honest, we found them to be somewhat more comparable than we had anticipated based upon our EPR results. So that was a little surprising, but good to validate pre-treatment the type of hypoxia status that you have for your tumors. Then we did the PO2 measurement with regular medical grade air and then changed to 100% O2. And then we irradiated with 10 gray and did the experiment again after the therapy. And here are the results right here. Well, if you use medical grade air and you measure the average PO2 within the 10 mice with the Colo 357 tumor, and then you look at the PO2 with medical grade air after radiation, you don't really see any difference. And the p-value is not statistically significant. There is a statistically significant difference when we use 100% O2 gas as the breathing gas, but it's more moderate at 0.031, not below 0.01 as we would really like to see. The SU8686 model behaved the same. There's actually an increase in the PO2 from medical grade air, which is counterintuitive. You'd think that there would be less oxygenation within the tumor after we've wrecked blood vessels with the radiotherapy. And again, p-value is not statistically significant for either of these cases, even with 100% gas. So a moderate, interesting biomarker, but not really going to get us there, right? But what about the difference? What if we look at the difference between 100% gas minus the medical grade air before therapy, and also 100% oxygen minus the PO2 with medical grade air after therapy? That's plotted here. And you can see that there's a very big difference between this difference prior to therapy and after therapy, P of less than 0.001. So that's a home run, right? And same for the other tumor model. In fact, perhaps even better. Again, SU8686 is supposed to be the more normoxic or more treatable model. So the results came out here to be a little more extreme than for the Colo 357, although wouldn't go so far to say that that proves that the therapy was better on this model. It's just an interesting thing. Okay, so what is this? What is delta PO2, right? This idea of percent or PO2 makes sense, the oxygen partial pressure. But what is the difference between these two gases? Well, I like this analogy. In fact, I'm going to skip this and I'll get back to it, where I'd like to show this physiological interpretation. So let's say you're driving down the road and you look at your gas tank and it's 1/4 tank full. That means that there's 3/4 of a tank that is empty in your gas tank in your car. In the same way, these tumors could be 1/4 tank full of oxygen under normal breathing gas, medical grade air. And then if we blow in as much oxygen, much gas as we can into the body of the mouse, well, that's going to fill up the gas tank, the O2 gas tank within the tumor, all the way to whatever full might be. And, certain tumors might have a very large amount of capacity, just like a truck gas tank, and other tumors might have small gas tanks like a motorcycle, right? But that doesn't matter. We're looking at the relative change of, how much extra capacity is in that tank at any given time. And so we're really wrecking the available unused oxygen gas capacity within the tumor when applying radiotherapy. And that goes down quite a lot for mice that are treated. They just can't absorb any more oxygen. There's metabolism and delivery going on under normal conditions. But then if we try to really blow in more oxygen, it's not going to work. We've wrecked that, right? All right, so another marker or another way of statistical analysis for evaluating this is to look at the effect size, which is the change before and after therapy divided by the average of the standard deviation of the measurements, right? And there are more sophisticated ways of looking at the combination of standard deviation. I'm just taking the average, which is pretty reasonable for this data. And you can see that the change is not much compared to the standard deviation. So the effect size for PO2 with medical grade air is actually rather poor. The 100% O2 data shows a better effect size, but of course, the best effect size is shown with this delta PO2. And an effect size greater than 0.8 is considered to be large, and it's very large for the delta PO2 and very large also for this other tumor model at 2.41. So again, another indication that, how much effect did this radiation have? Well, delta PO2 is the best biomarker to look at that effect. Okay. Well, as one other example in this realm of radiotherapy, we have also looked at TMX, also known as Manganoscan. It's a manganese oxide nanoparticle provided by another company. And when we use TMX to radiosensitize tumors, we actually see better tumor control after applying the radiotherapy, or in this case, immune checkpoint blockade. My apologies. So again, we're oxygenating the tumor, reducing hypoxia. Hypoxia often causes resistance to immunotherapy. So by lowering hypoxia, we've lowered that resistance and allowed immunotherapy to work better. So that's great. P of less than 0.05 between these groups. So that's great. And in particular, we have noticed that the hypoxic tumors became more normoxic when we used this TMX agent. And the normoxic tumors are ones that weren't terribly hypoxic. Well, they didn't change much at all. In fact, in this one particular case, it went down, but I think that's within our error bars. So pre-treatment to post-treatment for the hypoxic tumors, we see a significant change. And I think that's what's driving this data for the tumor survival here as well. And so for comparison to the MSOT, which is a great technique, but more indirect and looking at vascular hypoxia and understanding the tumor microenvironment, here we're actually looking directly at the tumor microenvironment, looking at PO2 between the blood vessels and the tumor cell wall. So I really like this technique for studying the effect of hypoxia on immunotherapy because those immune cells are in that compartment. And there are other radiosensitizers to be tested as well. All right. Well, I'd like to move on and describe some other research going on in the EPR community. I'd like to thank others, Boris Appel and Howard Halpern in this particular case. And you'll see names of other contributors here at the tops of these slides. And so I really do want to thank everyone who's contributed some of their research to describe other applications for EPR imaging. So here's a 3D map, isotropic of tumor PO2. And again, you can see a lot of blue here. So a lot of hypoxic regions within this tumor, but there are also some less hypoxic or closer to normoxic tumor regions as well. And that can be seen in this flashing picture right here with the red and the blue. That's showing the PO2 map. And you can see again that there's a part of this tumor that's quite hypoxic. and therefore should really receive a higher dose of radiotherapy. There should be dose painting based upon the PO2 map. Well, Boris and Howard did this study by making a cast that could go over the tumor that had a hole that matched the region of severe hypoxia. And then after applying radiotherapy, they would see a better tumor survival or better mouse survival in that study. They did the opposite experiment, not quite a control, but truly the opposite type of experiment, where they made a cast that would then hide the hypoxic region and expose the more normoxic region of this tumor region. And then they would irradiate. So they're irradiating only the normoxic sections, not so much the hypoxic section. And of course, tumor mouse survival went down quite a lot. So again, this idea of getting a reliable PO2 map, especially in real time, that could be then applied to the patient with radiotherapy right away, right there in the clinic, I think would be a home run, because then we could finally get to really outstanding dose painting based on PO2. As another biomarker, Dr. Halpern at Chicago has been discussing this for a number of years, that if you were to look at the number of or the fraction within the tumor that has very low O2, very low PO2 at less than 10 torr, that can be a predictive biomarker of the response to radiotherapy. Here's a case of a tumor outlined in red right here that has a lot of blue pixels, so it has very low PO2. In fact, here is the distribution. This is work with Martina Ellis back in the day in 2013 with Dr. Halpern, where they measured a substantial fraction of this tumor had PO2 less than 10 torr. And sure enough, those tumors that had less than 10 torr when they were treated actually had good survival. I'm sorry, those that had high hypoxic fraction, so it has a high hypoxic fraction less than 10 torr, those do not survive very well. The mice that actually do not have large, severely hypoxic regions within their tumor, they actually are treated quite well with radiotherapy. So again, if we could get that hypoxic fraction for patients, I think that would be a home run for determining which patients, to predict which patients would respond well to radiotherapy and which ones may not. Along with that same idea, Murali Krishna at the NIH has also been performing EPR imaging along with other imaging methods to look at the tumor microenvironment. Here's a case of radioimmunotherapy, combining the two with irradiation and anti-PD-1 antibody treatment for immune checkpoint blockade. And then to measure EPR at the end of the study, and here are the EPR maps of the PO2 for this tumor on the leg of the mouse. What I really want to focus on are these graphs here, looking at the combination of radiotherapy and immunotherapy together, showing that they had the greatest hypoxic or lowest amount of hypoxic fraction in these tumors. Again, that's the amount of tumor that had less than 10 millimeters of mercury, also known as torr. And the other therapies really didn't have as much of an effect of reducing that hypoxic fraction. That's different than measuring the median PO2, which in this case was a little more equivalent for these different tumor, different treatment types, and did not have statistical significance. So does this matter? Yes, again, the hypoxic fraction, day 0, when starting this treatment, seems to have some correlation with the tumor volume at day 9 in the study. So this could be used as, again, a predictive biomarker for our patients someday in the future. Martina Ellis, now in Krakow, Poland, has developed an oxygen-containing pegylated microbubble. This microbubble will then release oxygen through cavitation with ultrasound. And is this actually occurring in the tumor? Well, yes. With EPR imaging, you can measure the amount of oxygen that's in the tumor interacting with the free radical agent. So the free radical agent is not inside this microbubble to begin with, right? So this oxygen has been released into the tumor microenvironment. And you can see there's quite a burst effect with the microbubble and the ultrasound together relative to all of the control kinds of studies, right? And that can be done with an intravenous or intratumoral injection. Does this matter? Yes, you do get better tumor control. Tumor volume is lower with this treatment relative to control, corresponding again to the O2 release. Unfortunately, cavitation will cause an increase in metastases to lungs, as seen by these dark bars here. Some more work has to be done to avoid metastases of the primary tumor caused by ultrasound cavitation, but I think of that as more of an ultrasound effect. And of course, there's great research being done in that area. This is not related to the EPR imaging at all. But there is certainly tumor weight is a little better controlled with this therapy relative to control. So a great example of using EPR imaging to monitor the early response of O2 release in order to identify whether there's going to be a longer term outcome from this treatment. All right, so I'd like to switch over to a few other topics then for using EPR imaging. Marignane Kotecha and her colleagues at O2M Technologies have developed a very nice system for looking at in vitro cell cultures. So here's a plate of cells with different amounts of different cell density within each of these wells. And you can see that with no cells in the wells, you have high oxygen, as you would expect. If you have a very large number of cells, well, then you have low oxygen. Those cells are eating the oxygen pretty quickly, right? Consuming that a lot. That can be seen in this graph where as you increase the cell density, you are going to have less oxygenation. Well, why is that important? We tend to have incubators and the tissue engineering systems and so on that apply the same amount of oxygen on our samples. But of course, we know that different samples consume different amounts of oxygen. Now we have a way of actually monitoring that quite nicely using non-invasive imaging, allowing our cells and our tissues to grow. And we can then ensure that they're growing under well-oxygenated conditions, and we can boost the amount of oxygen as needed. In fact, I really like this example showing a system here where if you look at the top, middle, and bottom of this container here that has cells growing in it, well, the top has more oxygen than the middle, and the middle has more oxygen than the bottom. Well, seems intuitive because you're blowing oxygen in from the top, and the O2 output is also from the top. And so this system is really not being mixed as well as one would possibly hope. And that could then again change how your cells are growing or how your tissue is being engineered. So I think a fabulous non-invasive way of mapping out the O2 within a 3D volume. And really a clever application of this EPR technology. In fact, different cells have different consumption of oxygen as well, as you would expect. So can this be applied for other kinds of research? Well, I've been mentioning tissue engineering. Here's, I think, really an interesting example from the mealworm beetle larva. Just during lunch hour, you get to see a mealworm beetle. How's that, right? And so this is a bio-inspired material from this larva that actually helps to direct oxygen through the system, through these channels. And so does this work? Well, yes, using EPR imaging, you can follow the amount of oxygenation within one of these in vitro systems. And relative to control, there is faster oxygenation, more PO2 in the system. And at the very end, there's just a more total PO2 as well. And so this all can be monitored in real time, non-invasively. And I think it's a great technology for tissue engineering. All right. So can you measure anything else? All we've talked about is oxygen, right? I've pretty much sucked all the oxygen out of this presentation, right? Well, yes, there are other biomarkers that can be measured. Valery Kramtsov and his colleagues at West Virginia University have done a great job making a variety of EPR agents over the years to look at redox, glutathione, and so on. I'd like to focus on this set of agents here. To measure extracellular PO2, pH, and inorganic phosphate. And I think as time goes on, we've all seen the merits of doing multi-parametric molecular imaging. Multi-parametric MRI is now becoming more of a common term to take multiple MRI parameters together, right? Well, same here. Multi-parametric EPRI is possible, although matching the hardware with the type of agent still needs to be worked out some. So here's an example where the phosphonate of this agent can change its protonation state relative to pH. And therefore, the two peaks that give rise to these different forms in the EPR spectrum can be measured, and the relative ratio of those two peaks can then be used to measure pH. So that's based on the height. So as I've been mentioning, it's the T1 relaxation time, or also related to the width, in this case, the line width of this system, that tells you about PO2. And so this system, the free radical, is dependent on the PO2 in the system. Therefore, you get a nice linear correlation between PO2 and the line width that is dependent on relaxation properties. Then there's also a chemical reaction going on between the agent and the redox state of the agent relative to phosphate. If this is a very fast exchange rate, then the lines get very broad, and that can be modeled, and you can identify the frequency of exchange rate of this process, and that is related to the phosphate concentration. More phosphate, faster conversion rate you'll have here, right? And so you can get all three biomarkers at a single time, although I find it most interesting myself to consider pH and PO2 together because I'm studying those two biomarkers in the tumor microenvironment. Therefore, I'm just really thrilled that Valerie has shown correlations between PO2 and pH now within the normal mammary gland, not so much within spontaneous tumors, and tumor xenografts are just all over the place. So we like to say that hypoxia drives tumor acidosis, right? Somehow the HIF1 element and then promotes expression of all those proteins involved in lactate production and tumor acidosis. But I think this shows that, yeah, there is something there, but not all the time. And you really have to understand your system and see if there's a direct correlation or not, because sometimes two different metabolic processes don't always crosstalk to each other very strongly. So I think it's a wonderful result. So can we get this to work in the clinic? Well, the answer is yes. And here, Paranan Kuvasamy at Dartmouth, Coops, has developed a solid material, a powder, and it's actually a crystalline material that can be then ground into a powder type of material that has a lithium with an unpaired electron. In this crystal system, oxygen can pass through these channels and interact with the unpaired electron of the lithium. And we can play the same game of measuring T1 relaxation to get the PO2 from this powder material. And it has an advantage, and it's not in a liquid form, so it doesn't go anywhere when you put it into the tissue. And therefore, Coops and his colleagues are able to take a trocar, a needle that can then insert a solid material within tissue, and under ultrasound guidance, place this oxy chip material, in this case, more solid-like than, I shouldn't have called it a powder, perhaps. But by embedding it within a tumor, within a patient, in a surface accessible tumor, then the patient can be placed within an EPR instrument, and then a paddle can be placed onto the location of where that powder or where that oxy chip is located, and the PO2 can be measured. In this case, it is an EPR spectroscopy technique, so not doing imaging at this point. But it's still pretty amazing that we can get PO2 measurements from a solid tumor that is surface accessible. And so Coops has done a number of studies. You can see here a variety of patients to measure their amount of oxygenation at different statuses, and has really established safety and feasibility of EPR oximetry from a spectroscopy standpoint, shown variable levels of oxygen, and really highlighted the need for individualized measurements, because you can see on a per patient basis, these measurements are they're inconsistent from among the group, right? And in fact, that is not a detriment. I think it's really important for us as molecular imaging researchers to consider that we have to really evaluate individual mice and not just the whole group and take the average and be happy with our publications. Because frankly, patients don't care about the group average. They care about themselves, right? They don't want to be that 9 out of 10 mice survived, but the 10th one didn't. They don't want to be that 10th patient in the group, right? And so we really have to think more and more about individual basis measurements, in my opinion. And that's highlighted here, that we're getting individual patient PO2 maps or PO2 measurements, which is pretty remarkable. So is it feasible then to move this into imaging? After all, this is molecular imaging, not molecular spectroscopy, right? Well, yes, and I'm just really excited for our colleagues at O2M Technologies, along with their partners in this field, to create a human-sized 60-centimeter pole gap EPR imaging instrument with a resonator that will now fit a rabbit. They're not quite up to human size yet for the resonator, but it is feasible to get maps of PO2 within this rabbit tumor model, a very standard VX2 tumor model. You can see that there is a distribution of pixel or values here within the system. And quite a few of these pixels are below 10 Tor, that magic threshold that seems to be intriguing for future applications to predict radiotherapy. So I'm pretty excited that you can see a patient could fit between these poles someday. Of course, more development needs to be done with the resonators and pulse sequences and energy deposition and contrast agent approval and so on. But you could say the same thing 25 years ago about a lot of our MRI technologies or PET technologies, where others brought up that there were impediments to those technologies, and look where we are today with those other techniques. So I'm excited that someday EPR imaging will also be in our clinical radiology clinics based upon these initial steps. So with that, I would just like to thank those who I work with, including everybody in the research group who gets some of my crazy ideas to work once in a while, and also colleagues at O2M Technologies who have been wonderful to work with, other collaborators, and funding. And with that, I'd like to remind you that I'm excited to join the social lounge right after this presentation, where we can talk more about EPR imaging and tumor metabolism and radiotherapy and so on. So I'll join you in the social lounge. Thanks. Marignani, back to you.
Thanks, Marty. Such a big overview of EPR field overall. So we will stay here for another 15 minutes before we move to the social launch. And if you have question and answer or something to comment on the chat, I will share my screen. I believe, Marty, if you can stop sharing, I can share my screen showing the QA. So looks like people are still processing all the data you showed. This is my Q&A. And all, please ask your questions, and then we will move to social launch, I think after at 11, as far as I understand. So I had one question, Marty, about this delta PO2 measurement you were showing. So was it with medical air, or it's 100% breathing gas?
Well, both. And so we first start with medical air, and then we go to 100% O2 as a breathing gas and do our PO2 EPRI measurements with both gases. It's no longer needed to do the test retest, so we could shorten our scan time and time it appropriately to get agent into the tumor, even with an intravenous injection. I think a 15-minute scan window would be fine. So we could do the MRI before or after that, and do IV injections now. To kind of add to that question, I have gotten feedback that carbogen could perhaps be better than 100% O2. And I would agree with that. There's a less vasodilation with carbogen, and we're really wanting to study the effects of radiation on immunotherapy or on vascular disruption, not necessarily vasodilation. And I've done other studies where I've actually used 10% oxygen in the gas, in the breathing gas. By the way, that 10% oxygen is about the same amount of oxygen in the Himalayas near Mount Everest. So that's getting pretty low. And yet the mice can tolerate that, at least for a short period of time. And so you could think of actually modulating the amount of O2 in different ways to look at different effects. So we're doing that with flash radiation.
Oh, that's wonderful. Are there any questions from audience? We have quite a lot of people here.
To add just a little more information, I think it's intriguing to think of cross-validation as well. I think EPR imaging could be used in conjunction with BOLD and TOLD. We're already doing EPR MRI for an anatomical MRI. I'm intrigued to combine EPR and PET imaging, which has been done in Howard Halpern's lab at the University of Chicago. Could we investigate FMISO, FFASA, other agents, and use EPR imaging to help understand how accurate and precise those PET imaging agents are? So I think there's a field there as well.
Yes, sorry. Oh yeah, that will be very interesting. So still waiting to hear if there are questions from the audience. So basically, you don't need to probably, since you already know the difference between medical air, breathing air as well as 100%. So probably you, in future, we can rely on one because anyway you care.
Yes, very much. In fact, for many of our studies of radiosensitizers, we only use the medical air 21% O2. We want to see how much oxygen is going into the tumor based on the radiosensitizer. And therefore, there's no need to blow more oxygen into the tumor in that case. It's only when we look at the effects of radiotherapy or immunotherapy sometimes we want to see this delta PO2. And by the way, to be fair, I should point out that it's not like we invented the idea of oxygen enhanced. Certainly many years of research has gone into that for MRI and also photoacoustic imaging, that MSOT method. And like to give others a lot of credit, Ralph Mason and in particular has done that with MRI and MSOT for many years. We just repurposed that idea for EPR imaging, and it works out extremely well with the EPR because they're measuring PO2.
Okay, we have a question. So Mahadev Gorain asking, I have one general question about mice tumor detection. Is there any contrast agent used to detect the tumor in optical image or micro CT image?
Yeah, that's a great question. So I only showed that we use anatomical MRI for the registration with EPR imaging, and therefore we identify the tumor within the EPR signal map, right? While we can use a gadolinium-based MRI contrast agent to do contrast-enhanced MRI, and that would help to highlight where the tumor is, I would do that after the EPR because you don't want to be adding more metals to your system and then measure an EPR signal, right? It would just confound things. But you could do the MRI afterwards. Now, your question about micro CT is really intriguing. We actually do EPR and CT as well because of scheduling issues on our small animal MRI. That works fine for subcutaneous tumors that we can identify around the periphery of the body. Orthotopic is tough because there isn't enough contrast enhancement within the tissue with endogenous CT. Now, I haven't used a CT agent to enhance the tumor, but if you can find a differential contrast in the CT between the tumor tissue of interest and the other tissue that you want to ignore, well, yeah, it would be very easy to circle that with an ROI and map that ROI onto the EPR data, and no trouble at all. Same with optical imaging, although there, optical imaging is, pretty skin deep only, right? So I can't imagine how you'd get a full 3D image of the tumor using optical imaging, but if you could, and again, with fiducials, very easy to register 2 sets of images within a 0.5 millimeter resolution or error, and it'd be very easy then to find the tumor within the EPR image, just like your PET and other techniques that are used for anatomical with PET.
There's another question from Martina. Hi, Marty, great presentation. I'm wondering about the variability of delta PO2. Are there big differences between different tumor types or between the individual mice?
No, pretty consistent. And in fact, we've tried the COLA 357 and SU 8686 models in that same study with test retest. We're starting to get away from doing test retest now on other mouse models. And while I didn't show that data, it's pretty consistent, about 2.5, no more than 3 TOR for test retest repeatability. So I really like that with this instrument. And all the tumors were about the same size. So I really can't comment on tumors that are small or large or necrotic or highly vascularized. But of course, if you have a necrotic tumor, you're not going to get much agent in there. So you have to decide how much precision you're going to get there.
Okay, there is another question from Gabriela. She's asking about OXO71 propagation inside a solid tumors regarding their distorted vasculature. It's probably have some impact since she's imagined there is a less probe inside the tumor.
Yeah, I think a good way of thinking of this is that the sensitivity for EPR imaging is the same as the sensitivity of MRI. You're going to need the same amount of injection and so on. So if you can see contrast enhanced gadolinium-based MRI, you're going to see the EPR, no problem. But if it's below the detection limit of MRI, then yeah, I think EPR will it'll be difficult to see enough EPR agent. So again, necrotic tumors aren't going to take up much agent, and certainly like cartilage, right, has some difficulty getting gadolinium in there. Same with EPR. You wouldn't measure PO2 in cartilage, probably, right?
In my experience, we see more OXO71 gets accumulated in tumor.
Yeah.
And it doesn't go out because of poor vasculature.
Yeah. In fact, for those interested in PO2 of normal tissues, that's all above 40 TOR, maybe even above 50 TOR, except for the bladder. And the bladder is very hypoxic in our bodies right now. And that's a good reference organ to look at, because you will see good signal in the bladder. Of course, the agent is going to get to the bladder, and you're going to have low PO2 in there. So the signal is going to last a long time. So we like to see that bright blue bladder within our 3D images when we can.
More questions for Marty?
Well, if there aren't any, we could certainly start to switch over to the social lounge, if that would be a good plan. And that does take a couple minutes anyway, and we're near the end of the hour here.
Okay, I think that's a good plan. So
okay.
Let's see, everyone. Please join Social Launch. Here is the button on your screen, and I think we'll meet there.
Very
Thank
good.
you, everyone who are not joining us to Social Launch. Thanks again for joining for this webinar, and we hope to continue this series with more talks in the field.